This is a University of Otago podcast.
Kia ora everyone, and welcome to the second day of the Bite Size Science at the International New Zealand Science Festival. First up today, we have Amarni Thomas and Julia Horsfield from Pathology and talking about why we make glowing fish.
You can use a mic if you want.
Oh, okay. Is it working? No. It's not working. Yep, that sounds working. Good morning. My name's Julia Horsfield, and in my lab we look on solutions for new cures for leukemia, but we do it in rather a strange way in that we use a particular animal model to try and understand how leukemia genes work. And to give you some idea of how that happens, I'm going to hand over to my PhD student, Amarni Thomas, who's going to explain her project as to why we would want to make glowing fish. Over to you, Amarni.
Thanks. All right, so why did we make glowing fish? I'm hoping that by the end of this talk, we can all answer that question. So why would we be interested in blood? Well, blood is pretty awesome. So blood is really important for bringing the nutrients around your body and removing the waste. So what's in blood? Inside blood, we have different types of cells. So we have red blood cells. These guys are important for taking oxygen around your body and removing carbon dioxide. We have white blood cells. These guys are involved in helping fight infections, immunity, parasites. And then we also have platelets. And platelets are the things that clot your blood. So when you get a cut and you bleed and then eventually it stops and you get a wee scab, that's thanks to platelets. But blood cells aren't made in the blood, they're actually made somewhere else. They're actually made inside your bone marrow. So inside your bone is bone marrow. And in there's some really special cells. And these guys are kind of like starter cells. They're called stem cells. And these cells have the potential to become white blood cells, red blood cells, and platelets. So that's pretty cool. But how do they decide what sort of cell to become? And how are they able to do that? Inside these starter cells, they have genes. And this is how scientists typically represent a gene. So there's an arrow showing the start of a gene, where the cell will start reading the information. These grey boxes here is the information that the cell needs to become a different sort of cell. And then this black line here is called non-coding DNA, or what we originally thought as junk DNA. So the cell didn't actually need that in order to become a different type of cell. But what we're finding more and more is in this junk DNA, we're actually finding really important parts that could be acting as switches. So they could be involved in turning these genes on and off. And why would that be important? Well, in order to make all these different types of cells and know when they need to become those cells, the switches might be the thing that turns that gene on and off. And if those switches were to stop working, you could possibly get blood disorders. And in our lab in particular, we're interested in blood cancer. So in order to figure out how things can go wrong, we need to know how they work normally. And we do that in a pretty cool way. So we obviously can't study on humans, but in order to find out where these switches are, we can look at animals that also have blood. So we can look at mice and we can look at zebrafish, and we can look to see if they have genes similar to the human. And so here we see a really similar looking gene to that human gene. And we look to see what's kept between all of these animals in the junk DNA. So if it's kept, then it must have been important in evolution. So in our lab, we use zebrafish as our animal model, and these guys are super awesome. So zebrafish, you can actually look at under a microscope. And once the sperm has connected to the egg, that egg becomes fertilized. And we see a one cell. And then within 40 minutes, we can see 2 cells. Within a day and a half, we can see what looks a little bit more like a fish than this 2-cell stage. And in fact, At 2 days, we have a little zebrafish swimming around. And this one here at 4 days, and you can see it's starting to develop those fins, which is pretty cool. So how do we find out what these switches are doing? Well, we take the thing we think is a switch, put it with a glowing tag, and put it into the zebrafish. And then we look at those zebrafish under the microscope. And what we're seeing is that there appears to be some glowing where the switch is important. So whichever cells are using those switches will glow green. And it looks to be a very similar pattern here to what we see in normal zebrafish blood development. So what does zebrafish blood look like? Well, here we can see some zebrafish blood racing around, and this way it's coming back to the heart here. But what's happening in that glowing zebrafish? Well, in this glowing zebrafish, we can see the blood racing around the cells and coming back towards the heart. So those glowing cells are, in fact, blood cells. So that switch is important for blood. So why did we make glowing fish? We made glowing fish to help figure out how blood regulation happens normally. So once we can figure out what these switches are, where they are in the body, then we can hopefully help people who have blood disorders or blood cancer, because they might have mistakes in those switches. So thanks very much for listening, guys, and thanks to the people who sponsor this research as well, and thank you for the opportunity to talk today.
Awesome. Thanks very much for that. Does anyone have any questions? Cool, I have a question.
Okay.
How are you selecting or generating your tissue, or your potential tissue-specific primers, or promoters, I should say? So the little yellow boxes you were using, how are you picking out which ones, or potential?
So we look through the genomes of those different animals to see what's the same, and then we're actually cutting them out of the mouse DNA. So we're using the mouse DNA, or the switch in mouse DNA, to drive blood expression in zebrafish.
Cool.
So that's pretty cool.
Awesome. Thank you. Any other questions? No? All right, we'll leave it there. Thanks very much.
Cool. Thanks, guys.